To function as a precision optical instrument, the human eye relies on a stacked series of physical and chemical barriers. The first line of defense is the orbital cavity housing the globe. This physical armor is constructed from 7 distinct skeletal bones. The frontal, sphenoid, lacrimal are produced by the lacrimal gland, swept across the corneal surface, and actively routed through the upper and lower punctum into the lacrimal sac, before draining into the nasal cavity. Together, this solid skeletal protection and continuous fluid irrigation clear debris and absorb impact, securing the baseline required for visual processing. Zooming in on that fluid reveals it isn't uniform water. The tear film is a highly specific 3-layer chemical shield. The outermost lipid layer is secreted by the meibomian glands. This thin sheet of oil spreads across the surface to lock in moisture and prevent the underlying fluid from evaporating. This diagram illustrates the full stack. Beneath the lipid seal sits the middle aqueous layer produced by the lacrimal gland, followed by the innermost mucus layer from conjunctival goblet cells, which anchors the entire film to the corneal epithelium. Because each layer serves a distinct mechanical purpose, a blockage or failure in any one of these 3 specific gland sources instantly compromises the optical clarity of the eye. With the surface protected, the eye requires a mechanical engine to control its physical orientation. This is handled by 6 extraocular muscles. This schematic tracks their specific directional pull. The 4 rectus muscles, superior, inferior, medial, and lateral, manage primary horizontal and vertical movements. The remaining 2 oblique muscles handle torsional rotation. To maintain stereoscopic depth, this complex muscular yoke system must execute its movements with absolute bilateral symmetry between both eyes. Otherwise, the brain receives misaligned data, resulting in double vision. While the external surface of the cornea requires constant moisture, the inner layers demand the exact opposite. They must remain perfectly dehydrated to stay transparent. As seen in this cross-section, fluid naturally and passively leaks from the aqueous chamber into the corneal stroma. To counteract this, a fluid pump mechanism within the corneal endothelium actively pushes the liquid back out. This means the eye constantly expends metabolic energy for the strict purpose of maintaining its own baseline transparency. The deepest anatomical layer is the retina, which relies on a distinct nutritional environment that is tightly regulated. Here, vertical arrows show fluid flowing out of the highly permeable fenestrated choroidal capillaries, abruptly halted by the zonula occludens, the tight junctions of the retinal pigment epithelium. Sitting directly between these 2 circulatory environments is a thin structural dividing line known as Bruch's membrane. By strictly controlling what passes through, this microscopic barrier isolates the delicate photoreceptors from the constant unfiltered fluctuations of the body's systemic blood supply. To actively inspect these microscopic barriers in living patients, clinicians inject a water-soluble contrast agent called fluorescein dye directly into the bloodstream. This diagram explains the optical pathway used to capture it. The dye absorbs blue excitation light directed into the eye, and then emits a longer, yellow-green wavelength back through a barrier filter to the camera. On the angiogram, returning light maps the perifoveal capillary network, framing the dark avascular zone at the fovea center. Since dye molecules cannot cross intact RPE-tight junctions, the image remains clean. Any glowing leakage instantly pinpoints a physical failure in this anatomical barrier. From the orbital bones to the tight junctions of the RPE, these architectural barriers provide the mechanical and chemical stability required for the eye to function as a precision optical sensor.